Hello, my name is Hillary Rasnick, and I am a student at Southwest Virginia Community College, and I am going to be doing a presentation today on the bacterial cell, the structures inside and out. I have gotten all of my information from the textbook, Microbiology: A Systems Approach, 7th edition, and I have chosen to do my presentation on Chapter 4, the bacteria in RK. We're primarily talking about bacteria today, though. Um, and let me just say that there's so much more to bacteria, to a bacterial cell, than what I can describe in the time I have. There's so much more to it, and I've just decided to talk about or pick out or highlight the main things that I can, which I'm going to talk about the structures from the outside in, and I guess I'll just dive right into that. All right, the bacterial cell is made up of three different structures: the external layer, the boundary layer, and the internal material. The external layer is the first one I'm going to talk about. The external layer is made up of structures. These structures consist of appendages and surface layers. And the appendages are flagella, pili, fimbriae, I'm not sure how you pronounce that exactly, nanowires and nanotubes. All right, right here in this first little picture here, you can see a picture of the pili, and the second picture is with all this fuzzy hair around it is a cell with it's got the fimbriae sticking all off of it. All right, I'm going to talk about the flagella first, though. All right, the flagella of a cell is for motility. It provides a way for the cell to swim around in an aqueous environment. It's like the arms of, or like the tiny little propellers, like on a boat, but like arms that help swim, or a tail even, they help it swim through an aqueous environment. And set up in a polar arrangement, which means that the flagella could be at one end, or both ends, a bunch on one end, or all sticking all around it out of the cell. Monotrichous, amphitrichous, lophotrichous, and peritrichous right there in the diagram. You can see which one's which. Mono means one, which one tail. Amphitrichous got the two. Lophotrichous, a bunch from one end, and the peritrichous means that it's coming from all around. All right, and also the flagella, I want to say that what it does is it helps the cell swim to another cell. When that cell finds another cell, a DNA transfer is going to happen. And how this works is, okay, the fimbriae and the pili and the nanowires, all right, these are not for motility, are for attachment. The fimbriae is like, just like on this first diagram I've got here on this slide, this one looks like a hot dog weenie. It's got all kinds of hairs all over it, and those hairs are used for attachment, to help attach to other cells, and to surfaces. All right, so that fimbriae is going to grab on, the fibers are going to grab onto another cell. These are kind of like the fingers, maybe, to hold on to it. That's the way I like to look at it. To hold on to another cell, or like the bristles on a brush. But they're all over it, and they are mostly made of protein. But anyway, so they'll grab onto another cell after the flagella help the cell swim um, to another cell. All right, and I think my face may be obstructing the view of the one in the bottom corner here, but you can see this is the pilus down here with the blue at the very bottom diagram or picture. The pilus is sticking out, and or pilus, and it is holding on to another cell. That's the next slide here. It is a long appendage made of pilin. It's a special protein, and the made of a special protein called pilin. And the pilus is this long appendage, and it's like um, a sex organ, if you will, that is used for the purpose of conjugation. And that's when the pilus will hook onto and grab a hold of another cell, and it will act as the donor cell, and it makes a DNA transfer to the recipient cell. But yeah, the pills will actually bring or pull the recipient cell close enough to it so it can make that process of conjugation happen. Okay, and there's also, excuse me, let me go back here. Well, I can't go back, but there's nanowires also, and the nanowires is another extension or external structure. Excuse me, it's the extension of the cytoplasmic membrane. Um, um, and the bacteria use these to transfer amino acids to each other for food and energy. All right, we have the surface layers next. The surface layers of the bacterial cell. First layer is the S layer, and it is the protective armor-like layer of the bacterial cell. They tend to only show up when threatened, like in a hostile or harsh environment. I think that's really neat. That's when the only time that they're seen. But if the cell is in a calm, just good calm environment, it's not going to show up. Excuse me. So it's used for armor or for protection from threat. All right. And the next layer, the next surface layer, external surface layer, is the glycocalyx. It's a coating of polysaccharides. It's either a loose glycocalyx or a capsule, a thick-like structure glycocalyx. And the loose glycocalyx is a slime layer. It is more susceptible to being eliminated by antimicrobial drugs, to antibiotics, easily washed off. But over here, the capsule, the thick structure glycocalyx, that capsule is kind of like an armor. It protects pathogen from phagocytosis or being killed. It's less susceptible, less penetrable, because of that protective layer on it, kind of like the outside of a coconut. All right. Next, we have the boundary layer. Okay. Okay, the boundary layer um, um, of the bacterial cell. We've got the cell envelope and the cytoplasmic membrane and the outer membrane. All right, the cell envelope, it's where it's made up of, or excuse me, the cell envelope. We have the cell wall, and the cell wall gives shape and provides structure and support to the cell. And it's made up of peptidoglycan, and that, by the way, is the only place on Earth that it is found is in the cell wall, and I think that's awesome. Okay, and we have the cytoplasmic membrane, and that is a thin sheet that surrounds the cytoplasm. It is the that last fine sheet that goes right around it. All right, and the outer membrane, by the way, it's another part of the cell envelope. And all three of these are stacked upon one another, by the way, the cell wall, the cytoplasmic membrane, and the outer membrane. And let me go right over here and show you. Okay, in gram-positive and gram-negative cells, um, gram-negative, they have three layers. Gram-negative cells do. That's where the outer membrane is found, with the thin cell wall and the cytoplasmic membrane. Um, you've got the outer membrane, the thin cell wall, and then the cytoplasmic membrane. But over here on the gram-positive cells, a gram-positive cell only has two layers. It's just got a thick cell wall and the cytoplasmic membrane. And it's primarily made of, on the gram-positive bacteria, that it's the cell wall is made up of a big thick wall of peptidoglycan. Right. All right. Structures of bacteria. All right. Um, the internal structures that is the guts of the bacteria. Got a good little picture here of all of it right here of a cell and all the insides. The cytoplasm, that is the gelatinous or jelly-like substance inside of the cytoplasmic membrane, that last thin sheet that I was telling you about. It is what holds or includes ribosomes, inclusions, the cytoskeleton, and the endospores. The cytoplasm, that jelly substance, it is made up of salts, sugars, and amino acids, and it's also made up of 70 to 80% water. And that is a solvent that is used for the supply of molecules and used for nutrients and for the building blocks of the cell. So included in that are ribosomes, the site of protein synthesis. And ribosomes are used for translation. They translate, they convert language. The ribosomes convert the language of nucleic acid or RNA into the language of protein. All right, and the inclusions store nutrients, and that's where the food, the storage unit, the storage compartments for the food of the cell. And the cytoskeleton, it is within the cytoplasm, and it is made up of ribosomes, granules, and the fibers that act as that. That's what makes up the cytoskeleton in the bacteria that have them. All right, and I just want to make a note that not all bacteria are the same. All bacteria do have that cytoplasmic membrane, the nucleoid, the cytoplasm, the ribosomes, but some bacteria they don't. Some bacteria only some bacteria have that S layer or the fimbriae, those hairs, or that outer membrane, which would be the gram-negative bacteria, or the cell wall, the pilus or the flagellum. Only some bacteria have those, but all bacteria do have that cytoplasmic membrane, the ribosomes, the nucleoid, and the cytoplasm. But I hope that you did learn a little something from me today, and I hope you learned something about the bacterial cell. Uh, Thanks for listening.